of life, whereby this topic, what you're supposed to look at is DNA structure. Describe the structure of DNA. That's a question. Describe the structure of DNA. In this case, what are you supposed to look at? I'm going to tell you. Number 2, you know that DNA replication, yes. Transcription, translation, yes. These ones, you know them. It's easy to get them. But now, the most important part here is the structure of DNA, because sometimes people neglect that. You know how to describe DNA replication. Structure of DNA, how do you describe? Just in case it has come. First of all, you know that DNA is made up of a simple structure called nucleotides. If they ask you what is the monomer of DNA, you have to know that monomer of DNA is a nucleotide. And these nucleotides, yes, they have different parts, whereby this is a phosphate, this is a sugar, and this is a nitrogenous base. This is a phosphate, this is a sugar, is a nitrogenous base. Remember, this side you also have the same kind of structure. Yes? If you have the same structure this side, it means that if they ask you to describe, you have to tell us that DNA is made up of a nucleotide. Yes? These nucleotides are made up of phosphate. Nucleotide is a mark. Nucleotide is made up of phosphate, sugar, that is deoxyribose sugar, and nitrogenous bases is also another mark. Then you mention the nitrogenous bases. What are those nitrogenous bases? Then you tell us is adenine, is thymine, is guanine, and then is cytosine. You mentioning this is another mark. Then you tell us that it is made up of 2 strands. Yes, these strands, you telling us strands is another mark. These strands are joined together by weak hydrogen bonds. These weak hydrogen bonds is also another mark, weak hydrogen bonds. And then it twists around to form a double helix. Double helix is also another mark. There is only one mark I've not spoken about of you mentioning the bases, but also saying that whereby adenine goes with the thymine, guanine goes with the cytosine is also another mark. You mentioning that adenine, thymine, guanine, cytosine is a tick. Adenine with thymine, guanine with cytosine is also another tick. So that is DNA. So all these, if you count the marks which are there are 8 marks, but we only need a few of them. And those few of them, what is it? We only need 6 of them. So don't go to the paper without knowing to describe the structure of DNA, because it belongs to 6 marks. DNA replication. Here you have to describe the structure of DNA. DNA replication, whereby you tell us that DNA double helix is unwinds, weak hydrogen bond, break, yes. Unzip and the breaking of weak hydrogen bond is 1 mark. We don't give you 2 marks there, is 1 mark. Then to form 2 separate, let me repeat it. The word separate must be there. To form 2 separate strands. Each strand is acting as a template. It's Zoleka, I think you are hearing me. Ne? Yeah, I'm just looking at the people who are live, so that if you have any challenge, let me know in the comment section. I will answer it immediately. All right. Saying 2 separate strands is another mark. The 2 strands, they will act as a template. They will act as template. And then using free floating DNA nucleotide. We use DNA nucleotide to form a complementary strand, whereby Adenine goes with thymine. Guanine goes with cytosine. I see. Then you form this DNA with one original strand and one new strand, which are genetically identical. That's how you can describe DNA replication. But we have some few questions. I'm gonna show you these other questions because this is a 6-mark question. Just in case, remember your paper, it has almost 45 marks full of this short question. In each section, we bring at least one, of which this, at least one will come. All right. Then, transcription. Transcription must also know it, that DNA double helix unwind, weak hydrogen bond, break to form 2 separate strands. Now, one strand acts as a template. You have to know that. One strand acts as a template to form the messenger RNA using free floating messenger RNA nucleotide. In this case, we are using free floating messenger. For the case of DNA replication here, here we are talking about transcription. But when you're talking about DNA replication, 2 strands act as a template, while in transcription, 1 strand is acting as a template. Then, whereby adenine goes with uracil, adenine goes with uracil, and then guanine goes with cytosine. Yes? Then this messenger RNA moves out of the nucleus via the nuclear pore to the ribosome. So, that's, once you write it like that, obviously in these questions we always have questions or marks which are excess. So we are looking for only 5 to 6. Transcription is done. What about translation? In translation, depending on what you have memorized, but I'm going to show you the marking point in each. If you skip, you have lost a mark. According to messenger RNA codons, codon is a mark. The transfer RNA with the complementary anticodon, anticodon is a mark. Complementary anticodon is also another mark. Brings the required amino acid. It brings the required amino acid to the ribosome. The required amino acid is also another mark to the ribosome. Ribosome is another mark to form. The amino acids are joined together. The amino acids are joined together by peptide bond to form a required protein. That's it. Nothing else. According to the messenger RNA codons, the transfer RNA with the complementary anticodons bring the required amino acid to the ribosome. The amino acids are joined together by peptide bond to form a required protein. DNA code of life is done in terms of the 6 marks. So it means that you have at least one question which is most likely to come. Now listen, I'm gonna show you now these other questions which might come, or which you're gonna find in your paper. Listen. Before I go there, students make a mistake and they call peptide bond, they call them polypeptide. This is dumb. This is wrong. Polypeptide bond, we don't have this. We call it peptide bonds. We don't have polypeptide bond. Please don't call them polypeptide bond. Call them peptide bond. And then I'm going to show you also another mistake where students make. They call a polypeptide chain polypeptide chain a protein. It is only a polypeptide chain if it is less than 50 amino acids. 50 and above amino acids, we call them protein. It's only a protein when it's 50 amino acid and above. I hope you are getting it right. Okay. What we are saying here.
Recording in progress.
Sorry about that. Ne? All right. We are saying that. Look at the questions where other questions. DNA replication. Why does it occur? To increase the chromosome number. Why does it occur? To increase the chromosome number. Where does it occur? It occurs in the nucleus. When does it occur? It occurs during interphase. I think. How does it occur? Is the process which you have described. So DNA replication is done. Who discovered the structure of DNA? James Watson and Francis Crick. James Watson, Francis Crick. Another question. What is the difference between DNA nucleotide and RNA nucleotide? What is the difference between DNA nucleotide and RNA nucleotide? They are not asking what is the difference between DNA and RNA. They are looking for only the nucleotide of DNA and the nucleotide of RNA. So in this case, you talk about DNA has deoxyribose sugar, RNA has ribose sugar. DNA has DNA nucleotide has thymine, RNA has uracil. We don't have any other. That's it. Ne? Someone is asking me that I should repeat the structure of DNA. Describe the structure of DNA. Yes, I'm going to go to even genetics. I'm going to go even to evolution. I'm going to show you the whole paper, what you expect, and within these 2 hours, you'll be able to write the paper. Ne? You write the paper within 1 hour. So the discussion we're going to do within these 2 hours, you'll be able to write this paper within 1 hour. Meaning that nothing we're going to miss. Okay. So now as I'm winding up, what is the difference between the difference between DNA and RNA? Those ones you know. But there is one question they can ask you. What is the difference between transcription and transcription and DNA replication? Transcription, here you form one strand will act as a template, while DNA replication, the 2 strands will act as a template. I think. In transcription, yes, we use uracil, adenine, thymine, guanine, uracil. While in translation we use adenine, in transcription we use adenine, guanine, cytosine, uracil for the new strand which is formed, meaning that we form a messenger RNA. While on DNA replication we form DNA. Then in transcription, yes, you'll find out that in transcription and DNA replication, we have also some few similarities, whereby DNA double helix unwind, also in transcription, DNA double helix unwind. Weak hydrogen bond break, also decide weak hydrogen bond break. If just in case they are looking for the similarities there, you are able to answer that. Now, as I'm winding up this DNA, there is a question which is always a challenge to students. What is that question? That question is about the genes. The gene mutation. Gene mutation. Now, let me give you an example. If you have A, G, C, and this is codes for leucine, and you have A, C, G here, and this codes for tryptophan, yes, and then you have A, C, G, and this also codes for leucine. Then they ask you, how will the protein be affected if this is changed to this? Because you form a different amino acid, the protein functioning or the functioning of the protein will change. So it means that you have to tell us that when AGC is changed to ACG, tryptophan will be added to the protein instead of leucine or the polypeptide chain. This leads to a different protein or the different functioning of a protein. You mentioning this and mentioning this is a tick, and also mentioning different protein is also another tick, because this question is always 3 marks. What about if this with this G changes this and this changes to this, even if it is 1, but it is the same amino acid? This means that the functioning of the protein will not change. However much the base sequence has changed, but what is formed codes for the same amino acid. Therefore, it means that you will find out that it is exactly the same protein. The functioning will not change. It's like someone saying that, for example, let me just use an example here. Someone can call you Sibusiso. Someone, you will say yes sir, or can call you Ntuli. Still you say yes sir. I think, meaning that is the same kind of person. But if this, because this is leucine and this is leucine, what if someone calls Sibusiso, and then the next time the next caller calls you Dobe? Obviously Sibusiso will not reply. No, Sibusiso will not reply. It's Dube who will say that yes sir, because now it is different protein, the functioning will change, but it's the same protein, the function will remain the same. So I repeat, I've concluded with DNA. We will only look at it when you're talking about the questions. So here you're saying that DNA structure, replication, transcription, translation. Please don't go without knowing it. And this gene mutation of DNA. When you talk about DNA profiling, that one is a little bit easy. Please just read the advantages, disadvantages, and sometimes the trick which is there is they tell you that other than that what is mentioned in the question, give another, and you find the student giving the same answer in that question. So it means that you lose a mark. Let's check and see here. I'm not going to go through objectives, but I'll go through the terminologies because I love the terminologies. Here is the question of DNA. Let me just reduce it a bit. Now, fast. Now, this is just a simple question. What is X? In most cases, people say that X is a cell membrane. X is not a cell membrane. X is a nuclear membrane because the cell membrane is out. Nuclear membrane, this is a nucleus, but they can ask you the process. What is this? This is translation. Once you see something like this, it is transferrin A, this is a amino acid. But if they put dots there, this is not a protein, it's a polypeptide chain, because I said that polypeptide chain is less than 50 amino acid, above 50 amino acid, it is a protein. Since this is just 1, 2, 3, 4, 5, only 5 amino acid, therefore that is a polypeptide chain. And then what is this? This is messenger RNA, and then which organelle is this? Is a ribosome. Now, question can come. How many bases, how many nucleotides, how many codes code for amino acid? So let's answer that. How many bases code for an amino acid? Amino acid, remember, an amino acid, yes, an amino acid, yes, it's always having 3 bases because of this 1, 2, 3. So if how many bases is 1, 2, 3? But they're saying that if there are 3 amino acids, how many bases are there? So it's gonna be 1, 2, 3 times 3, which gives you 9. If I have 9 bases, how many codons are there? Remember, each 1, 2, 3 is one codon. So how many codons are in 9 bases? So we'll say that divide by 3. Therefore, how many codons? Codons will be also 3. I think you have to know when to divide and when to multiply. Yeah. Lastly, on this, they can also give you a question that they can ask you a question that, look, they can ask you a question that 20% is adenine. What is the percentage of guanine? So you have to know this. Ne? If 20%, you have to know that A goes with T, C goes with G. Ne? C goes with what? C goes with G. So if A is 20, this one is also what? 20. So 20 plus 20 is equal to 40. Ne? So now, because this percentage is out of 100, therefore I'm going to say 100 minus 40. So it gives me 60. I think if it is 60, what is really happening there? 60 is going to be for these two. So divide by 2. So it's going to be 30 is going to be 30. So it means that the percentage of guanine is going to be 30%. Don't go to the paper without knowing how to calculate the base. Ne? Yeah. I'm using these as examples. So we are saying that which organelle is X? We have seen organelle X is nucleus. We have seen it. Let me do like this. Is nucleus. And then which molecule is Z? Which molecule is Z? Where is Z? Where Z is messenger RNA. Don't just say RNA. It's messenger RNA. State 2 locations in the locations of DNA in the cell other than the nucleus in the cell. So it means that you have to talk about number 1, mitochondria and chloroplast. Yes, mitochondria and chloroplast. Describe process W. W is we don't have any other process occurring in the nucleus except transcription. So you have to describe transcription. I've exhausted most of the questions you're gonna find in DNA Code of Life. It's only one last question. Let me yes, this is the question, how to interpret this. And however, I've tried to explain something here. The trick behind here is one. How? The trick which is here, please check and see what is there. If they say this is DNA and they gave you DNA at the beginning, then you don't need to change. If they say they give you DNA and what is here is transfer RNA, you have to change it to messenger RNA and you have to change it to transfer RNA. Are you seeing? So, in this case, they are saying that name the DNA-based triplet code for serine. Now this is transfer RNA. Serine is here. So I'm going to say is UCG. This is transfer RNA. Convert it to messenger, which is going to be, yes, AGC. Then convert it to DNA because they want DNA. How many times do you convert? It depends on what they have given you. So it's going to be, yes, it's gonna be A, yes, G, and then C. So that's gonna be, are they saying serine? Yes, that's what is gonna be the answer. First 2 amino acids coded for Z in the diagram. So they are saying they want to identify. Now, this is transfer RNA, but Z, what is Z? Z is messenger. Therefore, you have to just change this into transfer RNA, one conversion, because it is messenger to transfer RNA, and then you come to the table, you find it. What is the change in sequence of nitrogenous bases? I've talked about it here, that these changes. Gene mutation, sudden change in the sequence of DNA. So that's what you call gene, or the nitrogenous bases. When they change the sequence, then automatically you're gonna have a different protein structure if it codes for a different amino acid. So what is a change in a sequence of nitrogenous base in DNA code? Gene mutation or mutation. They're saying that code CUU changes to C. Now they're saying codons. It means that they are not saying anticodons, codons. So it means that I have to convert this into transfer RNA. Sibosiso, are you hearing? All right. We are saying that change this is gonna be GAA, and then this is gonna be GGA. So GAA is gonna be what? So now you saying this is gonna be leucine, like the same example I gave. CCU is gonna be GGA. GGA is gonna be what? Proline leucine. Now you writing to us that this is gonna code for leucine, this is code for proline. Those are 2 marks. Leucine a mark, proline a mark. I think now you tell us because of a different amino acid is coded for, it will result in a different protein. But if it was coding for the same amino acid, then the protein would have remained the same. So it would have said that this code is for the same amino acid, for example, leucine, and also this code is for the same amino acid, which is leucine. This will not change the functioning or the structure of the protein. But because now it is changing, this is leucine and then this is proline, you're mentioning leucine a mark, proline a mark, this will result in a different protein or functioning of the protein. All right, let's go to meiosis. Yes, meiosis, unless you have any question. Someone is asking me, where do I get, where do you get my booklet? There is a link in the description below. Ne? Yes, that link, it will take you to the website. You go to grade 7 to 12. Then it's going to bring you another page which has past paper 2022. It's going to bring you notes. You click on notes. Then it's going to give you the different subjects for grade 12 and grade 11 and stuff. You click grade 12 and then it's going to give you the subjects now. Now the subject, when you click life science, is going to show you the different topics. Then you download the topic per topic. Yes. That's how you can get that booklet. Let's look at meiosis. Meiosis. What do you expect to find in meiosis in your exam? Meiosis. What do you expect to? I'm now talking about the 6-mark question. I showed you the 6-mark question for DNA. So now I'm showing you the 6-mark question for meiosis. You cannot miss. You will come back and then comment that, sir, thank you for giving us the scope. Yeah. If you follow what I'm telling you, I guarantee you a distinction. Share the video to others so that the others can also. Someone is saying that it's not clear. The clear is as a result of your the internet and your internet connection. You can increase the maybe from 128 to maybe 480, something like that. All right, let's continue. Meiosis, number 1, you need to know how to describe the karyotype. Karyotype. I think you know the karyotype. If you don't know, I'll show you how to describe it. You need to know how to describe crossing over. You need to know how to describe non-disjunction. Ne? Now, this one is a star, is a drawing. Phase before, phase after, phase at this side. So you have to know that. How do you describe a karyotype? Let me see if I can find a question here which has a karyotype. And then I'm gonna describe for you so that when I'm describing, you see né? You see what I'm trying to describe. Sorry for, yeah, here is an example of a karyotype. Describe the karyotype of individual 2. Ne? But when the question comes, might not, the karyotype might not be there. You have to be knowing it by head. For example, the karyotype is individuals are made up of 46 chromosomes or 23 pairs of chromosomes. Yes? From you mentioning that 46, ne? 46 is a mark. Ne? 40, sorry. You know, technology. What's happening? Okay, let me check and see. 46 or 23 pairs. Don't say 23, because if you just say 23 without writing the word pair is wrong. It's made up of 23 pairs of chromosome. From pair number 1, yes, to pair number 22 is called orosome. You see now we are describing a karyotype. It's called what? Orosome. Yes, the 23rd pair is gonosome or sex chromosome. Yes, the 23rd pair is made up of X and Y. If X and X, they're together, the individual is a female. All those are marks. If X and Y come together, the individual is a male. You see, you are describing the karyotype. You have already exited the 6 marks. Each chromosome pair, they have different sizes. So that's how you can describe the karyotype. It's made up of 23 pairs. Pair number 1 to pair number 22 is called orosome. The 23rd pair is called a gonosome. These pairs, yes, each pair of chromosome have different size. The 23rd pair, if X is made up of X and Y, if X and X meet, is the individual is a female. Is X and Y meet, the individual is a male. That's how you describe. Now, what about if they bring a karyotype like this? Yes, like this, and then ask you which one is a male, which one is a female. Check. This is short. This is, 2 of them are long. Therefore, this one is a male, this one is a female. Give a reason for your answer. The chromosomes here, they are not of the same size. I see here, same size and shape. While here, they are same size and shape. So it means that this is X and X, while this is X and Y. Remember, we said is longer than Y. That's why you find that to happen like that. Now, someone is asking me, all right, I think it's sorted. Now, we are saying that, let's go to another question of karyotype here, just in case, because I was describing the 6-mark question, but there can bring some few questions here. What are somatic cells? Somatic cells, these are body cells. Ne? These are just body cells. Name the specific type of chromosome number 1 to, I said it is the orosome. Each pair shown in the homologous chromosome, each pair, state the origin of each pair of chromosome. Definitely, one came from the nucleus of the ovum, another one came from the nucleus of the sperm. Meaning that one chromosome, for example, this chromosome came from the mother, and this chromosome came from the father. So came from the nucleus of the ovum, this one came from the nucleus of the sperm, and then fertilization took place, and then a zygote was formed. That's what they are looking for. They are saying that state 3 characteristics of chromosomes which are homologous. Homologous chromosomes, these are chromosomes with the same shape, size. same location of genes. Not same genes. No. Same location of genes. If they are same genes, then it would have looked alike. But the chromosome where they are located, the locus of these genes in these chromosomes are the same. I think now I'm clear on that. So please, you must not miss out any question. Explain one observable reason why these two individuals are not identical twins. Why are these ones not identical twins using this karyotype? These are not identical twins. Why? Because this is a male and this is a female. Obviously, you know that if it is a male, what happens? What happens? If it is a male, the sperm is gonna be Y from the father. Ne? And if it is a female, the sperm is gonna be, if it is a female, the sperm is gonna be X. You understand? We only have one ovum. So I cannot use one sperm and then I form X and Y. Never. Now, why are they not identical? Because of the different sex. Observable, the 23rd pair, this individual one has X and Y, while individual 2 has X and X. So, identical twins are as a result of one sperm fuses with the ovum and then the ovum splits into 2. Non-identical twins are as a result of 2 sperms fusing with the 2 ova. So, meaning that you have different individuals. That's why is the non-identical, you can have a boy and a girl, but the identical twins is either boys or is girls, because we used one sperm. If the one sperm of X fuses with the ovum, then it's gonna be identical twins girls. If one sperm of Y fuses, it's gonna become identical twins male. So that's exactly happening. Yes, I think yes, I'm done with the karyotype. What about crossing over? Crossing over. Describe the process of crossing over. I think I organized for you a question which you're gonna see in crossing over. Oh, I think I have, it's not there, it's here. Crossing over. Yalla, it's here. When you talk about crossing over, you talk about 2 homologous chromosomes. These chromosomes, they come together. I think these chromosomes, they come together. They cross over at a point called chiasma. They must be homologous. The word homologous chromosomes align together. That's a key point. Homologous chromosome. They come together and the chromatids of the homologous chromosome cross over at a point called chiasma. I think at a point called chiasma. You understand? And then these chromosomes, these chromatids, they exchange the genetic material. When they exchange the genetic material, yes, they split and then they form genetically different chromosomes. That's why I told you these are genetically different chromosomes. But these homologous chromosomes, they are not the same. They don't have the same chromosome number. They have the same location of the genes in their chromosomes. That is the meaning. All right. Now, what is P? What is this P? We have talked about, we said that easy. Some people, they call this centromere. That is not a centromere. A centromere is Q. Please don't confuse centromere and chiasma. C, chias, H, this pen, chiasma. If one is called chiasma, if the men we call them chiasmara. This is a centromere. Centromere. And then this is a chromatid. It's not a chromosome, it's a chromatid. What about this? These are homologous chromosomes. Sometimes they can ask you, how do we call the homologous chromosomes under crossing over? It's called a bivalent. It's called a what? A bivalent. Chromosomes under, homologous chromosomes under crossing crossover. We call it a bivalent. Yes, I've not checked if because the schedule changed. If just in case that electricity goes at 4 o'clock, ne, I will come back at 6 o'clock live. But if it doesn't go, we are still continuing until a distinction is scored. Yes. All right, let's continue. Now, we are done with the crossing over. Let's talk about non-disjunction. What is non-disjunction? Are you there, guys? Non-disjunction. If you have any challenge, if I'm going to another point, please let me know. I'll read your comment there, and then I'll come back to you. All right. Non-disjunction. What is non-disjunction? Before I go to non-disjunction, where does crossover take place? It takes place during prophase 1. Don't just say prophase. We will get it wrong. Prophase 1. Phase 1. What are some of the causes of mutations? No. What are some of causes of variation? We'll talk about them. Causes of variation. I'll see how do people lose marks here. You think that you have written the paper, can't the paper has written you? I'll show you. There is no way the paper is going to write you tomorrow. You're going to have to write it. All right, let's continue. God willing. We are saying that non-disjunction occurs during anaphase. Non-disjunction occurs during anaphase. It can be anaphase 1 or it can be anaphase 2. Anaphase 1 or it can be anaphase 2. So what is this non-disjunction? Check here. I'm going to show you how. Someone is asking me that, can I please repeat bivalent or bivalent? Bivalent is when homologous chromosomes are under crossing over. Chromosomes under crossing over is called bivalent or bivalent. Yes, that's what I said. All right, now come back here. You have chromosomes in metaphase. Sometimes students fail to understand the chromosomes under metaphase. The chromosomes must be in the equator. Equator is an imaginary line. An imaginary line. We just imagine a line which is happening. Yes? It's called what? Equator. But we say that is the center of the cell. So these chromosomes must be at the center of the cell. This thing must be on the sides. Unfortunately, I have not used the camera. I would have showed you. You'd have seen me when I'm trying to say sides. So that, but you have to know that, make sure that the centrioles are inside, so that you, sometimes we twist it so that we confuse you, so that you go out, so that we know that you if you just cram, then gone. So they must be on sides. So they can ask you, draw a cell after, cell before, or crossing non-disjunction. How is this non-disjunction? How this cell gonna lead to non-disjunction? Now, if these spindles contract and they pull this chromosome, now this whole set is pulled to this side, and then this one comes this side, this one comes this side. You result in a cell whereby this cell has 1 plus this, and then this one, because the whole thing went to the because this one went this side and the whole thing of this went to this side. Let me show you. Let me do like this. Let me do like this. Let me do like this. Let me do like that, so that, yeah. So it means that this part came to this side. This one came this side, while this one came to this side. It means that now this cell is going to have 1, 2, 3 chromosomes. What about this side? It's going to have only 1. I think. Now this one is 3 instead of 2, and then this one is 1 instead of 2. I think so. Because it has 1, therefore is going to call it monosome. Well, because this one has 3 instead of 2, it's going to be called trisome. I think. How does this lead to Down syndrome? How does this lead to Down's syndrome? I think. So it means that now, if this fertilizes the ovum, then it's gonna lead to an individual less of 1 chromosome. If this fertilizes the ovum, it's gonna lead to an individual with an extra chromosome, and this is called Down syndrome. But it's not called Down syndrome except it has occurred on chromosome number 21. So which chromosome number is chromosome number 21? Sometimes when we bring these karyotypes, okay, I think I used it. No. Karyotype. Where is karyotype? Sometimes, okay, I wanted to show you. Sometimes we bring it and then we confuse you on the karyotype, ne? And then we ask you, how did yes, check here, ne? On this chromosome number, they put another one there, and then ask you, is this chromosome individual fine? How many chromosomes? Yeah, how many chromosomes are there? Because you know that this an individual is supposed to have 23 pairs. When I just say 23 pairs or 46 chromosomes, wrong. Check before you conclude anything. Please check the chromosome number 21. I'm done with that. Let's go to this. They're saying that a drawing, we have shown it, but a drawing, it must have a title. Ne? Must have a title. When they ask you to shade, for example, if the chromosome is like this, and this one is shaded, ne? Please, when you draw, still draw it like that and it's shaded. If you shade it down, shade it down. If you shade it up, shade it up. After that, label. Have some 2 labels. I want to give you one thing students always mess up with. Is metaphase. Listen carefully. Don't go away. Anaphase. Yes, then telophase. I'm going to say this metaphase 2, anaphase 2, telophase 2. When these chromosomes, let me show you here down. When these chromosomes, look, I have it here. When they're under metaphase, we call them chromosome. When they're under, they're being pulled now, ne? They're being pulled to opposite poles, ne? Like this. These are not chromosomes. These are now chromatids. But the moment they reach the poles, we don't call them chromatids. We call them single-stranded chromosomes. Unreplicated chromosomes or single-stranded chromosomes. We only call them chromatids if they are at under anaphase. They are being pulled. Here, they are chromosome. These are replicated chromosomes or double-stranded chromosomes. These ones are called chromatids. The moment they reach the pole in telophase, it becomes what? It becomes unreplicated chromosome. Don't call them chromatid. Yes. Let's go back to this last question of meiosis. Then we conclude it. Yes. Okay, now they are saying that drawing variation. What is variation? Variation is the difference among organisms of the same species. Variation is the difference among organisms of the same species. Don't compare mangoes and oranges. Don't compare dogs and pigs. That's not variation. Variation must be comparing dogs. Dogs. Variation is the difference among organisms of the same species. It must be the same species. Yes. Now, what are some of the causes of variation? If you're comparing humans, you must compare humans. So what are some of the causes of variation? The causes of variation is, now, the major mistake students do in this exam, if they ask you 3 causes of variation, the first thing the student is going to say that the first cause of variation or genetic variation is going to say that meiosis. Yeah, we shall say correct. And then the next point is going to say crossing over. We're going to say thank you. Then it's going to say random arrangement of chromosomes during metaphase. We're going to say yes, but no. Why? Why are we crossing it when it's correct? It's because you mentioned here meiosis, and meiosis involves crossing over and random arrangement of chromosomes. So if they ask you this question, please remove meiosis. Don't think about meiosis. Then you talk about crossing over, random arrangement of chromosomes. There are 4. We will not put you this. We will give you a tick here and we'll give you a tick here. So you see, you have got 2 marks. Then plus you can talk about mutation, random fertilization, unless they have said that give 3 causes of variation during meiosis or during the process of meiosis. Then now you talk about crossing over, talk about random arrangement of chromosome, and then you can talk about mutation. Then you don't talk about random fertilization, don't talk about random fertilization or random mating. You don't talk about it. Yes. Yeah, I think that is the meiosis. Can we check and see? There is one question you usually lose marks. How many number of chromosomes? That question is always killing students. Is always killing students. Is always killing students. All right. Let's go through this question paper. This one. Then they're saying that what is A? A is a cell membrane, not nuclear membrane. You can't see nuclear membrane when a phase is meiosis, is metaphase. Spindle fibers, then this is a chromosome. Yes? Identify the phase of meiosis above. Because they are single chromosomes, therefore it's gonna become what? It's gonna become metaphase, because they are single metaphase 2. If they are double, this becomes metaphase 1. Metaphase 1. But here, single metaphase 2. All right. Let's go to another question. They are saying that what is A? We have seen it. What is B? We have seen it. Give one observable reason for your answer. Chromosomes align along the equator singly, individually. That's, if you don't say singly, 0. Or individually, 0. It must have that word, individually or singly. Chromosomes align along the equator, 1 mark, singly. What about describe the role of B in the movement of these chromosomes during meiosis. B is spindle fibers. They pull the chromosome, the chromatids, in this case, the chromatids to opposite poles. They try to shorten. When they shorten, they pull the chromatids to opposite poles. Charlotte, are you still here? All right, let's continue. Draw it. This is the question I was talking about. You can't go without. Put the title. Good. After putting the title, what next? What next? What next? You talk about you draw, because they're saying that draw a diagram of C, structure C. They're saying structure C, ne, as it would appear in the final phase of this meiotic division. They are saying only C. So if they're saying structure C, they're not saying draw the cell, how it will appear. Since they're saying structure C, then automatically you're gonna draw this. Okay, let's see. Structure C. You have two options. Why two options? Then this chromosome, you see, it's 1. So you draw that chromosome with the centromere there. While you can draw this one also, draw the same thing with the centromere there. Are you seeing? But now this one is shaded. This one is not shaded. Why? Because it is colorless. See? Nothing shaded. This one is shaded. Therefore, you shade it up. I see? When you shade it up, what next? So it's gonna be like this. But now, where am I going to get the marks? Remember, they're saying final stage. Final stage has a nucleus. So this chromosome must be in the nucleus. Don't forget that at the final stage, you have the centrioles. You also put the centrioles in there. Centrioles are outside the nucleus. Then after that, please give a title and then you label cell membrane, nuclear membrane. Now, okay, you can talk about centromere, and then now, this is not a chromatid. This is a chromosome. The chromosome I talked about, that these are called unreplicated chromosomes. These are unreplicated chromosomes. Yes. So I think you understand it very well. I think you really understand it very well. No, I think you got it right now. Ne? If you have not got it right, please let me know. Let me know. All right. Now, let's go to another sub-topic. Hey, there is one which stresses you. How many chromosomes will be there at the beginning of this cell? How many chromosomes are here? Oh, how many chromosomes are in this cell? So now, people always confuse. Please, if they don't mention this is a human cell, don't think about 46 chromosomes. Unless they have said this is a human cell. If they have not said this is a human cell, no, you are not supposed to draw both. You only have to draw one, because they say draw a diagram. Label the diagram, not diagrams. Ne? Someone is asking, should we draw two? No, we only draw one. Yes. Or follow the instruction. What is it saying? But in most cases, they want one. But when you're marking, we draw the two, and then we see which one to give you. Ne? Basically, depending on which one you have drawn on the memorandum, the answer is there, then we will do that. Yes. All right. Now, and when you are marking, we don't just mark. We just have to mark the title. We mark the title. We mark the shading. The shading is correct, maybe. Then we mark maybe 2 labels. Or the correct diagram, 1 label, and then the correct diagram, then here. All right. They're saying that how many chromosomes are found here in this cell? Someone is going to say 23. I ask you a question. 23 from where? This is not 23. I see 2 chromosomes. Why do you say 23? So it's going to be 2 chromosomes. People are always talking about 23, 23. Why? How many chromosomes will there be at the end of this cell in telophase 2? There will be 2, because this is meiosis 2. The meiosis 2 maintains the number of chromosomes. Another question. Sorry. Then they're asking you, how many chromosomes were there? How many chromosomes were there in the same phase of meiosis 1? There were 4, because meiosis 1 halves the chromosome number. So if meiosis 1 is 5, then meiosis 2 is gonna be if meiosis 1 is 10, meiosis 1 is if meiosis 1 is 10, meiosis 1 is gonna be meiosis 2 is gonna be 5. So there is half the chromosome number. So please, you only if they say this is a human cell. Sometimes they ask you that how many chromosomes will there be present in the same phase as a human cell. Now here, because it's meiosis 2, then you talk about 23. But if they don't talk about human, please and please, I beg you, don't talk about 23. All right. Then they are saying that lastly about meiosis before I go to genetics. I show you the 6 marks which are going to come from genetics, and then I show you the simple questions, the way we are trying to define them. Share the video to others so that they don't miss out. Don't forget to subscribe, like, and also drop the question which you really want me to sort out. All right. Question: what is the importance of meiosis? What is meiosis? What is the importance of meiosis? The reason why we have meiosis is to have half the chromosome number, so that this doubling effect, because you remember sperm, let me show you sperm. The sperm, we'll call it sperm, ne? All right. The sperm, what happens? This technology, Ibo. Okay, I hope it's right now. The sperm will bring 23, and then the ovum will also bring 23. So when they fuse during my fertilization, you form 46. Yes. So meiosis must divide them again so that they form 23. So that's why we are saying that it reduces the chromosome number, or it halves the chromosome number, or it solves the problem of the doubling effect of fertilization. Number one, that is it. Number two, the major importance of meiosis is to produce gametes. Gametes, these are the sperms and ova or ovum. Number three, where does meiosis take place? Where does meiosis take place? Meiosis takes place in testis, in human, and then in ovary, that is female. Anthers in plants, that is male, and the ovary, that is female for the case of plants. You have to know those things. I think I've exhausted the questions of meiosis, unless someone is saying that load shedding is coming. Oh, sorry. And then okay, can I make it downloadable? I cannot make it downloadable until it is I'm offline. The moment I'm done, I'm completely done, then it will be able to this one is asking me that how do I know the number I must count? The number is shown there. If look at this, these are 1, 2, 3. Let me check here. Okay, let me look for another question here. You will see the numbers. Sorry, when I'm looking for the question. Okay, that one. This one we are done with it. What about this? Let me check here. How do I know the number? How do I know the number? Oh, in this case we don't have, but this is the crossing over. This is the thing I was talking about, the process of crossing over. Centromere, chromosome, chiasma. This is a bivalent still. It's also a bivalent still. Also, this is a bivalent. But depends. You see now the crossing over here. There are two. So when they are drawing, for example, you have to draw them. In this case, you draw them up. You draw them up here. You draw them 2 times. So I think I've exhausted. But what you see, if the cell is showing you chromosome 1, chromosome 2, chromosome 3 like this, and then they ask you how many chromosomes are there. These are the number of cells, number of chromosomes, 1, 2, 3. So tell us 3. How many will be there in meiosis 1 of the same phase? There will be, because meiosis 2 is single strand, chromosomes are 1, 1, therefore it's gonna be 6 in meiosis 2. Unless they say that how many will be there in the same phase of meiosis, but for human. I say that if they don't talk about human, forget about. You see now? Here is the question. Oh, where in the human female body would this type of cell division? This is where does meiosis take place in female? I've answered this, and I say that ovary. Give a letter name of the structure that attached to the spindle fiber, which is C, and then this case is the centromere. How many chromosomes will be found in each letter cell at the end of meiosis? Now come back here and see. The diagram represents the chromosome in the cell that is undergoing normal cell division. You see, it's undergoing normal cell division. So now they're asking, how many chromosomes will there be found in each daughter cell at the end of this cell division? How many chromosomes? How many chromosomes are you seeing here? This is chromosome 1, chromosome 2, chromosome 3, chromosome 4, chromosome 5, chromosome 6. Because they are still under meiosis 1, so at the end of this cell division, it will be half the chromosome number, whereby 1, 2, 3, 4, 5, 6. So it means that the answer is going to be 3. I think Chantel, I think I've answered your answer correctly. Yes. Please share the video so that others can also benefit. Sometimes I do these lives, and then after that they look at it and they say, I didn't see this live. So share it so that even other people can benefit from this live. All right. Let's go to our favorite topic called genetics. Called what? Genetics. Share the video so that everyone can get all the marks in genetics. Genetics. I'm not explaining too much of this, but I'm explaining only what you expect in exam. I'm not explaining the basics. 6-mark questions. 6-mark questions. 6-mark question. When you talk about 6-mark questions, yes, in genetics, what are you supposed to look at? Number 1, genetic cross. In this genetic cross, you need to know, when do I say sex-linked and when do I say non-sex-linked? Yes, sex-linked and non-sexy-linked. I think load shading is not there. I think so. On my side, ne? I think it's not there. Ne? I think they have changed the schedule. All right. Non-sexy linked. Sexy linked. Ne? All right. Number 3 is pedigree. You have to know the pedigree, but still here, you have to know whether it's sex or none. Another thing, disaster, blood group. And then lastly, cloning. Here we are going to talk about biotech. All these questions are 6 marks. 6 marks. I'm not saying this is what I'm going to talk about only. These are questions which come with the 6 marks. With the 6 marks. So if you mess them up, you lose. As I said that each section you're going to have one which has 5 marks, 6 marks, 7 marks. Latest, the worst is 6 marks. This, please don't lose them. Don't lose these marks. Distinction. Call your name Mr. and Mrs. Distinction. Tomorrow we have to crash the paper. All right, let's continue. Genetic cross. But before I start genetic cross, I've not talked about terminologies. I've not talked about them. But I want to know this. What is a locus? What is a locus? Or a position of a gene along a chromosome is called locus. Now, when do I use sex-linked and when don't I use sex-linked? I'm trying to go through this. After that, I go back. I'll show you. When you're doing the genetic cross, guys, even if you don't know anything, just write P1 space F1. We shall come and give you a tick. So you can't go to the paper without having this. So you have gone to the paper when you know. When you say genetic cross, before even thinking P1 F1, you get a tick. Okay, this is a phenotype. Phenotype is always given in the question. They will say that blue eyes, married, black eyes. You see, you come here and say blue eyes crossed with black eyes. That's a mark. Another mark you have got. After that, you say genotype. This is the only disaster which is there. We're going to look at it. And then now, what about here? Now we say phenotype. We are done. Genotype, we are done. Then now you go to meiosis. Meiosis will only have meiosis if you have the gametes. For example, if I have T. Okay, sorry. I think some people are not seeing there. Let me just draw it here. They are saying that, okay, P1, F1. I'm going to show you when do you write P2. Ne? Because sometimes, yeah, but you saying P1, F1, mark. They say phenotype. Yeah, phenotype is always given in the question. As I told you that they can say big head crossed with small head. And then they produce a baby who is a big head. You see? I don't know. Are they big head or a small head? I don't know. All right. Then they are saying that big head crossed with small head is given in the paper. So you don't need to stress. So you get a mark for this. You get a mark for this. Now, after that, you go to genotype. This is the only disaster I've talked about. We're going to look at it. You have to identify it. I will show you. For example, T, small t, T, small t. Now you go to meiosis. Yes. They're saying meiosis. They're saying T, small t, T, small t. Which are these are gametes. You see? Now, gametes, in most cases, we love it when you circle them. You know why? Because sometimes people try to bring this close together. And once you bring them together like this, we cross. We don't give you a tick. So make sure that I prefer, always I tell my students, since you are my student now, circle them. Circle them. The moment you circle them, at least we shall know that, we shall know that nothing, even if they are too close, since you have circled them, still we will give you a mark. Now, then after that, you talk about fertilization, where you have the crosses. Now, you mentioning meiosis and fertilization is another mark. In most cases, this question is 6 marks. So you have got 1 mark, 2 marks, 3 marks. So you have got 50 out of how much? Out of 6. 50% you have got it. Before whether stress have started or not, whether you got these things correct or wrong, as long as you have that, you have the 3 marks. All right. Now, you continue. You cross this. Okay. I'm gonna show you another way how you lose marks, né? And how to benefit get marks. This T with the T, and then this with this T with the T, this with this T with the T, then this with this small t with the small t. Now these are offsprings. Listen, in the question sometimes we put what you call compulsory mark. We put what you call compulsory mark. When you're talking about compulsory mark, someone is asking me if I'm gonna do cloning. I'm coming there to cloning. Compulsory mark, for example, if this is a compulsory mark, even if you get 1, 2, 3, so it's 1, 2, 3, 4, 5, 6, and if this question is 5 marks and they say this is a compulsory mark, even if you say 1, 2, 3, 4, 5, 6, you got 6, but you lost the compulsory mark. We won't give you 6, no, we'll give you 4. We'll give you 4. But if you write this, the 3, the other 3, they are obvious whether the compulsory mark is there, whether the compulsory mark is not there. Those 3 are secured. You go to the paper when you have secured them. Then the other ones, the compulsory mark depends on the question they have asked you. In most cases, they ask you, show this. Then you do what? You show. That showing will lead to the compulsory mark. I hope I'm clear on that. Then they are saying, I'm gonna fire you a few questions here. So the crossing, all of them, they do the same. Oh, I have not spoken about determination of sex. Sex. Don't forget to stay virgin. Determination of sex. Determination. Someone is asking. All right. Sex determination. Someone is talking about meiosis. Meiosis, we finished it. Sex determination. Okay. But you'll be able to watch it when the live is done. All right. We are saying that question can come, and this always kills the student. How many genes control blood group? How many genes control blood group? Let me know in the comments. Just now. Just type how many genes control blood group. I'm waiting before I continue. How many genes control blood group? Let me see. Who is the first one to answer? How many genes control blood group? I'm waiting. Define mutations. Yes, I'll talk about the mutations. How many genes control blood group? Yes, I'll cover every topic. Don't worry. I'll cover every topic. How many genes? Guys, type. I want to see the answer. How many genes control blood group? I'm not seeing the answer. 4. Someone saying 4. Thank you. How many genes control blood group? I'm still waiting for the answer. Type. Another one saying, I think 4. All right, thank you. How many genes control blood group? Tell me, Chantel. Yeah, okay, I think 4. Thank you. I want to know. How many genes control blood group? 3. How many genes? Okay, guys. Thank you very much. That's what I wanted to know. Yeah. Now, since we are looking for the distinction, they are not 4. It's only 1 gene. The gene, it is 1 gene polyallele. 1 gene. Which gene is that? Blood type. Blood group is only 1 gene. Are we together? How many alleles control blood group? There are 3. 1 gene. Remember, which gene is that? Let me show you here. Yeah, Terence said after giving. Okay. Okay. Blood group. Blood group. Blood group The gene is called, it's like saying height. Ne? You're either tall or short. Is one gene which is height. You see? Height. You're either tall, height, you're either tall or short. You understand? Since it's one gene of height, but two alleles of tall and short. So blood group, the gene is blood group or type. Yes. How many alleles? There are 3. So say genes for blood group is 1. Alleles, there are 3. That is allele for A, allele for B, and allele for O. Sorry, allele for O. Like that. Are we together? Yes. Thank you for those people who tried to participate to give the answers. Since we are looking for the distinction, we can't go without knowing this. Gene, one gene. How many alleles? 3 alleles. Ne? 3 alleles. How many genotypes? Genotypes, there are 6. Yes? Genotypes, there are 6. You have this. Remember, A is either homozygous or heterozygous. That's 2. B is either homozygous or heterozygous. Then you have, that's 1, 2, 3, 4. Then you have A, B, and then you have that for O. That's it. So 3 genes, 1 gene, 3 alleles, 3 genotypes, and then phenotypes, there are 4, which is A, B, and O. So basically that is it. Oh, AB, yes, AB is codominant. Yeah, O is the this is homozygous. Ne? This is the codominant or heterozygous. No, codominant. This is heterozygous, is homozygous, heterozygous, homozygous. So there are 6 of them. 1 gene, 3 alleles, 6 genotypes, 4 what? 4 phenotypes. Now, a tricky question can come. A man who is blood group A married a lady who is blood group B, and they produced a kid of blood group O. Show how this is possible. A man of blood group A married a woman of blood group B, and they produced a woman of blood group O. How is this possible? It means that once you find that question, for you to cross, it means that a man, because they are saying they produce a kid of blood group O. O is small I, small I. Before I go there, let me show you the mistake here you do. And we don't mark it. However much you love it, we don't mark it. Some people, they write blood group like this. A. What is this? This is not I. Don't write I like this. And you put a dot like that. I is like this. Don't write I like this. This is 1 in Roman figures. Check. You have sometimes, you have watches, where it shows 1, 2, 3. Yes, they are like that. So this is not I. This is 1, this is 2, this is 3 in Roman numerals or Roman figures. So don't write A like this. We won't give you a mark. However much some computers, yes, they do. You are not a computer. Write the way
use
correctly.
my distinction material. Yes. Those people who use my distinction material,
make sure that you got all these are simple things which might lead you to lose the distinction. For those people who have been using my book, Distinction Material, you know exactly what I spoke about it, and those people who have been watching the video. So it doesn't matter now. What matters here is for tomorrow's paper, for you to crush it. All right. Now, we say that, how is a man of blood group A marrying a lady of blood group B and they produce a kid of blood group O? One, know that blood group O, it has two eyes. Yes? Yes. If they have two eyes, how about this? Remember, A can be homozygous or it can be heterozygous. B can be homozygous and heterozygous. Now, if I have O, which is II, yes, it means that I used this and this. Because if I make this and this, there is no way I'm going to produce these two. Even if I say this and this, there is no way. I'm going to only have one I, small I. Therefore, it's going to be heterozygous blood group A. So when you talk about the genotype, it's going to be the phenotype. So I'm going to say P1F1. Genotype, phenotype, phenotype. I'm going to say A crossed with B. And then I say genotype, I'm going to say A heterozygous crossing with B heterozygous. Then meiosis will take place. Some people, they then say A, I, B, this is B, ne? I. And then I circle them. Please don't put a cross here. Please don't. You'll get it 0. You'll get 0 there. Please don't put a cross there. Ne? Yes. Someone is asking, why didn't you find the channel at the beginning? That's the answer for God. All right. Now, let's continue, guys. But you can tell your grade 11 or your sisters, your brothers, subscribe the channel. Look for M.SAIDI. Yeah. You always find the content there. All right. Now, look. Now, this is gametes. Gametes. So, if these are gametes, now, what happens here? You can use a Punnett square method, or you can just cross. Whatever you want, still it is fine. For example, let me use the Punnett square because I've been using the cross. So you draw your Punnett square here, and then you say, okay, you have A, you have I here. You have B, you have I here, small I. And then now you cross, you're going to say this is A, B. Ne? This is going to be B. And then this is going to be A. And then this is going to be O. You see? These are offsprings. You see? Now, this one produced a person who is blood group O. That's how it was possible. It can even come in the 20 in the 7-mark question. That's why I showed you those crosses. So please, blood group should not be a problem. Which blood group is now I'm done with the long question of blood group. Few questions now. Which blood group is a universal donor and which blood group is a universal recipient? Blood group AB receives from every blood group. Well, blood group O donates. So blood group O is a universal donor. Some people, when they are blood group O, they think their blood is strong, they feel good. No, it's worse. Why? Because if you lose blood, if you need blood, we have to look for only one person with blood group O. But if your blood group AB, you are lucky. Anybody who comes, you can donate that blood to you. So blood group AB becomes a universal recipient. It receives from everybody. While blood group O is a universal donor, only donates to anyone. Someone is asking me that why these crossings are not allowed. The crossings are not allowed here because they are not crossing. They're already gametes. After that, you can draw your crosses here. Ne? So the crossing here, the cross here, no, it's not allowed there. It's not allowed. Crossing here is fine. If you cross them, it's fine. You can either use the Punnett square or you can just do the crossing. All right. Let's go to we talked about the blood group. I'm coming back with the cloning, cloning. Or I'll give you one tip of an example of, okay, let me give you the question, because I just want to go just for 2 or 3 minutes. Yes, 3 minutes. Let me give you just one question for you to practice for me. Is 6 marks, ne? Yes, this one. It's a simple question. There is something behind it, most especially on this question. I need to see so that you don't lose that mark. Please do that for me, ne? Do that for me. Then after that, I'm gonna do it again for you. What's the time? Is 39. I'm giving you 4 minutes. 4 minutes to finish, ne? I will be back. I will be back. I'm saying I will be back. I hope you are done, eh? Hope you are done. Hope you are done. Okay. Oh, come on, saying yeah, it's done. All right. Anyway, ne? All right. Okay. That's nice. Okay, now let's go through it. They are saying diagram below, okay, controlled by R and R. Check here. The first point is R and W. These are capital letters. The moment they are capital letters, it means that both of them are dominant. All right. Now they're saying red cross with white produce pink. You see, they have 3 phenotypes. The moment you have 3 phenotypes, it means that that is incomplete. Dominancy. It is incomplete dominancy. Incomplete dominancy. The question is here. State the type of dominancy shown in the Snapdragon plant. Here is incomplete dominancy. Give a reason for your answer, because a third phenotype is given or an intermediate is given. You see, a third phenotype is given or an intermediate is given. Now, if it is complete, if it was complete, ne? I would have been with red and white. I either produce red or white. Which one is dominant? Depending on which one is dominant. And then if I have codominance, red and white, I would have produced a mixture of red and white. But because this one is pink, a third phenotype, therefore, or an intermediate, therefore it is incomplete dominancy. They're saying Gardner crossed 2 pink flowered snapdragon plants using the genetic cross. Show the ratio of the expressed phenotypes in the offsprings. Now, remember I told you that the first thing you need to do is to write P1 F1. But in this case, they're saying they crossed they crossed two pink. Pink is found on the second generation. Therefore, it won't be P1, F1. It will be P2, F2. In most cases, students lose that mark. Ne? If it is the second generation, then you say P2, F2. If the first generation, then you say P1, F1. Ne? Yes. So now, there you talk about phenotype, which is pink. I told you that this is obtained from the question. Check. They are saying, what are they saying? Check what are they saying. They're saying genetic cross showed a gardener crossed two pink flowered snapdragon plants is always obtained from there. Are you seeing? Then now, so it means that I've got a tick here. I've got a tick there, and also I've got a tick there. So now 2 marks. Now they are saying that pink, remember they say that white is capital W and red is capital W, capital R, white is capital W. Therefore pink is gonna be capital W, capital R, capital W, whatever, capital W, capital R, or capital R, capital W. It's the same thing. So it's gonna be capital R, capital W, crossing with capital R, capital W. But this is the genotype. See, now you get another tick. Then they're saying that meiosis will take place, and then you have capital R, capital W, capital R, capital W. I said don't leave these so-called gametes to be as if they're at the beach. No beach, ne? At the beach, ne? You live naked, ne? You enjoy yourself. Ne? Don't leave them naked. Try to dress them up. Try to dress them what? Try to dress them up. At the beach? So try to dress them up so that, ne? Yeah, they are not there at the waters there when they are enjoying their life. Ne? Try to dress them up so that you separate them. Even if they are too close to one another, you won't lose a mark for that. You won't. Then after that, you do the crossing, or you do a Punnett square. So cross with R, capital R, capital R, capital W, capital R, capital W. Because I started with R, I will start with R, capital W, and then capital W, capital W. Whereby these are offsprings. Those are offsprings. Now, if those are offsprings, what is it? R, this is red, this is pink, and then this is white. Meaning that I will have one red, 1, 2 pink, and then 1, 1 white. There is a mistake where the student do, whereby they ask you the ratio. In this case, you write 1 to 2 to 1. Ne? Some people, when they ask them the ratio, they write expression, and sometimes when they ask you the expression, they write the percentage. So when they ask you the ratio, please write the ratio. If they ask you the egg, how many were red? How many were red? Or what is the percentage of individual plants which were red? Then you have to say it's 1 out of what is the total. The total is 4 times 100. Then you give us the answer. You give us the answer as 25%. Please don't forget that. All right. Let's continue. And we say our question is I think I've exhausted that. Pedigree, let's just look at one question here which involves the pedigree, and then we can go to our there is one question here for blood group. There's one question here for blood group, but I think we exhausted the blood group. But you see that blood group also has 5 marks. Explain why paternity of the boy could not be established using the blood group. Why is paternity test is not able to be established, like to conclude that this is the father of the individual? It's because many people have the same type of blood group. So you can't say that, as I showed you, that is blood group A married blood group B, and then produce blood group O, still they produce blood group AB, still they produce blood group B. So whom are you going to say that is not the father, who is, or whom are you going to say that is the father? So it means that you end up losing the what? The mark. All right. Why is it not conclusive? Why is it not conclusive? Because many people have the same type of blood group. But if they ask you, explain why paternity test for this situation, you will say that the mother is blood group O. You see, the mother is blood group O, while the boy is blood group AB, blood group B. So of which the boy can be can be B, heterozygous, or the boy can't be homozygous. Why? Because the mother is II, so gets 1 from the mother. So the genotype of the boy is heterozygous. Now this one can give yes the B, ne? And also this one can give the B. So whom are you going to say is the father? Whom are you going to say is not the father? So that's why in this case blood group can only help you to eliminate those possibilities which cannot happen. For example, a person of blood group, the father can't be blood group A because this person is blood group B. You see? So if it's blood group A, there is no way how the boy is going to have blood group B. So if there is a man, another man who is blood group B, so blood group A, that one is wiped out. You understand? And then another man who is blood group O cannot be. So blood group A and blood group O, they are wiped out. Why? Because if it is small I, the boy is blood group B. So there is no way how this boy is gonna get a B from the father who is blood group O, yet the mother is already known is blood group O. You see? So that's the meaning that you can't use blood group to conclude the father of the individual. Because your blood group B, you can't say that this is my child because this child of mine is blood group B. There are so many people who have the same type of blood group, and the possibilities of having these are many. I hope you understand me better. Now, how do you write this? It means that this one is only blood group A and blood group O are going to only be wiped out. So the blood group A and blood group O, they cannot be the father. So you get those 2 marks already. And then this blood group, the male A can give this allele for B to the boy. So it can be the father. Or this one also can also give 1 allele for B and gets another one from here. Also can be the what? Can be the father. That's why we cannot conclude using blood group who is the father of this boy. As I repeat that, blood group can only be used to eliminate the possibilities which cannot happen in a situation. Who is the biological father of the boy using the DNA profile? Now you have to check and see. You will see that it is this and this. You will see that is male what? Male 2. Give the reason is because the remaining bands of DNA profile match with the male. The remaining, they must match. If there is one which is not matching, then automatically it's not the father. Might be a relative, but might not be the what? The father. Remember, all the genes that this boy or this baby is having is mother and the father. So if you take out all the mother's genes, because the mother is known, now the remaining bands of this boy must be those ones of the father. Let me show you what I'm trying to say. This mother, okay, this one is not for them. Yes, this one is corresponding. So This one is out. This one corresponds. This thing is now jamming. All right, corresponds. This one corresponds. Oh, this one corresponds. Yes. This one is not matching. So this one is not matching. This one is not matching. This one corresponds. Is out. This one is not matching. This one is not matching because this is not. This one is matching. So now all of those ones which have X must match with the male who is the father. That's why I'm saying that the remaining bands of the boy, after those ones of mother have been removed, they must match with the father. Okay, this one, yes, it matches. You see, this one, yes, it matches. You see, this one is matching with this. This one is matching this. That's why you say that individual 2 is the father. Male 2 is the father of the boy, because the remaining bands of the boy have matched with the male male 2. All right. Now let's go to just one example of a pedigree diagram. Yeah, pedigree, where are you? Pedigree, and then I show you how you can use it to answer and also to score a mark. They are saying hemophilia is a that statement is enough. This statement, hemophilia is a sex-linked trait. Hemophilia is a sex-linked what? Trait. Meaning that if it is a sex-linked trait, what is it? It means what does it mean? It means that you're gonna use X and Y. Please, if they don't say is a sex-linked in exam, don't use X and Y. You're gonna get it wrong. Is a sex-linked disorder. Don't use X and Y if it is not mentioned in the question. So here it is mentioned sex-linked. Or sometimes they don't say sex-linked. They say that hemophilia, the gene is carried on X chromosome. Still that is sex-linked. If it's not mentioned, please don't even bother. Don't even waste your time. Don't even waste your time to let me show you here. One of the deafness in human is carried on a single allele. The diagram below shows, yeah, done. They are not talking about sex-linked. However much they talk about Paul, and you know that Paul is a boy, Liz is a girl, Mary is a girl, John is a boy. However much you know that, please don't use X and Y. Please don't use X and Y. So if they don't mention is sex-linked, don't use X and Y. If they mention sex-linked, then you talk about X and Y. How many generations are here? There are 3. 1, 2, 3. They are saying that, and then number 2, it can be caused by, something can be caused by a recessive allele, or it can be caused by a dominant allele. Anyway, let's continue with this question. They are saying, state what is represented by a circle or a diagram. I wanted to tell you that sometimes we don't bring that a circle is a male. A box is a male. A circle is a female. Sometimes don't give you that key. You have to go to the exam when you know that. All right. Now they're saying, what are they saying? Okay. Now they are saying, this is a boy. This is a. How many individuals? Let me brainstorm you. This is affected with hemophilia. Don't say is sick. Is sick of what? Sick of what? Malaria, ne? Sick of what? Sick of hemophilia, or is hemophilic. All right. How many kids are produced by P and Q? They are how many? It's 1, 2, 3, 4, 5. I know many people are gonna say that. It's not. They are not 5. 1, 2, 3, 4, 5. No. This one is an intruder. This one intruder. This one got married. This one got married. This. So there are only 3 kids. How many generations are here? 1, 2, 3. There are 3 generations. You understand? What is the phenotype, the genotype, or the phenotype? They can ask you what is the genotype or phenotype of P or Q, whatever they can ask. But if it is shaded, it means that that individual is affected. Now, they are saying that state what is represented by a circle? We have said that the circle means a male. State the number of general A. We have talked about them. There are 3 generations. 1, 2, 3. Offsprings P and Q. State the number of offspring P. The same thing is this, and this. This one and this ones, they are only and only got married to that. So they are intruders. State, they're saying that let us give the letters of only male who are having hemophilia. Letters of the female. Female is T and the female is U. Those are the only 2 people who are having hemophilia. Why? Because they are shaded. And then lastly, the genotype of R. The genotype of R. Now, how do we find the genotype of R? Look at the babies or look at the parents. First, look at the babies. It can help you. If you see that the baby is, one baby is hemophilic, then automatically the mother is gonna be heterozygous. So the mother is gonna, remember that here you have X and Y. Y doesn't carry anything. Y doesn't carry anything. So it's X and Y. So Y does not carry anything at all. All right. I think now, okay, so since this one is sick, it means that, and is a boy, so it's going to be like this. It's going to be, this is a recessive allele. So if it's a recessive allele, obviously this can't come from the father because the father already gave the what? The Y. Therefore, this came from the mother. So it means that the mother, since is normal but a carrier. So the mother is going to be, or R is going to be capital A, capital X, capital X, capital H, small h. And that becomes the answer for that. Yes. Let's continue now. Let's continue. So we are done with this. We are done with this. Sex determination and biotechnology. How do I explain sex determination? Sex determination. Sex determination is like describing a karyotype. When asked you to describe sex determination, it's like describing a karyotype. But here, if they ask you the genetic cross, you have to do the cross. Ne? So, for example, if it's a cross, you tell us that P1, F1, everything, ne? Male crossing with a female, yeah, and then you tell us that male is X. You tell us the phenotype here, ne? And then you tell us the genotype. Male is X, Y, and then female is XX. Then you cross. Then you tell us meiosis here. You tell us gametes. They come out here. They come out, and then I'm gonna explain. I'm gonna show you how to write it if they say using a genetic cross, and then also show you how do you describe if they ask you to describe. Then these are gametes. These are gametes. Then they cross, then they cross, then this and this, cross Y, and then this and this, they cross like that. So this is the offsprings, meaning that you have 50% chance of having boys, and 50% chance of having girls. What about those who have girls, or boys? Yes, even if you have a boy here, the next time you have 50% of a boy again. Even if you have another boy, the next generation, you have the next reproductive cycle, you're gonna have still 50% chance of having a what? Having a boy. So it depends on how do you roll your dice and how many chances do you have to have a boy. There are 50, but you are just lucky that you have what? Boys, or you are lucky to have what? To have girls. So in this case, we are saying that 50 boys, 50 girls. Yeah. If they ask you to describe, you tell us that individuals, humans are made up of 46 chromosomes. Ne? The 23rd pair is called gonosome. It's either X and X or X and Y. if X and X, if a male sperm with X fertilizes the female ovum, it will result in X and X. The individual will be a female. If a male sperm having Y fertilizes the ovum, remember ovum is always X, the individual will be X and Y, and therefore will be a male. There are 50% chance of having a male and 50% chance of having a female. That's how you can describe sexy linked. So let's finish up the cloning, and then we go to evolution. We finalize it with the evolution. All right. Biotech. Cloning. What is stem cells? What are stem cells? Stem cells, these are cells which have ability to divide to form other body structures. Imagine you are just a sperm. You are just a sperm, ne? Sperm fused with the ovum. You formed a zygote. The zygote, remember this paper one formed a ball of cells, ne? Which called the morula. You see, a morula is a stem cell. Because here, if I ask you, show me the eye, show me the legs, show me the chest, show me the, you can't show me. It's just a ball of cells. So these cells, they have ability to divide again, to form other body structures. You understand? Yeah, you form other body what? Structures. And then, yes, you become, yes, you have the eyes, you have the ears, you have everything. So we call them stem cells. Those are cells which have the ability to divide to form other body structures. All right. Give me 2 examples of stem cells. Umbilical, adipose tissue, bone marrows. So you have to know those stem cells. Sometimes we bring this as an extract. Let me check and see. Let me check and see if I have any question regarding to stem cells. This is gene. Calculate the percentage of, okay. This is gene. Oh, I skipped something which is very important. That is the dihybrid cross. Yes. Let me leave it there so that I don't forget again. Dihybrid. I want to show you how do you identify the genotype. How do you identify the genotypes? Using the gametes, because that is a very problematic question. Okay. So in this case, I can't find the question there. All right. Now, let's go to cloning. I only want to discuss about cloning here. Ne? Yes? About the what? IA. Cloning. Yes? I have sheep A. I have sheep B. Ne? I get a cell of sheep A from the skin. Let me say somatic. And then this one I get it from ovum. Ne? What I do, I remove the nucleus of this. I go with the nucleus. Ne? I throw away this shell. Well, here, I go with the shell, but I throw away the nucleus. I fuse them using electrical method, and then I form a cell which looks like a zygote. It looks like a zygote. Now, this zygote, I put it into another animal. A foster mother. Animal. Yes? Yes. And then what I do, I produce a lot of these animals, and these animals are identical. Question: why are all these animals identical? They're identical because when this forms a zygote, it forms a ball of cells. Now this ball of cells, now I divide these balls. Now I'll have small balls, and then the small balls I keep on going and then insert them in different other shape. Ne? So it means that because I use the same genetic material, obviously these ones are going to be identical. It's because it's the same genetic material which was used. Now, a question: will these babies look like B or will look like A? They will look like B. Why? Because I use the nucleus of B. I didn't use the nucleus of A. Why did I use the nucleus, the shell or the casing of B? It's because I wanted this to look like a zygote, because what is outside is an ovum. You understand? It's like drinking alcohol in a cup. I won't say that you're drinking alcohol. I will say that you're drinking tea. So because the outside is a casing of the nucleus, therefore this will look like a zygote. And because this one is diploid, 2N, they can ask you how many chromosomes are found in this. Because it's a somatic cell. Somatic cells are always diploid. You see? Somatic cells are always diploid. Someone is asking me to do the part of evolution. I'm coming there. I'm finalizing genetics here. And then I'm coming to evolution. So because this one is what? Is a somatic cell. Therefore, automatically, this is going to be a diploid. And then, because it's brought in the a casing of the ovum, so it's going to look like a what? A zygote. And now I split them in two small pieces, and then I insert them in different shape, and each shape will produce identical what? Identical shape. Yes, I think I'm done there. Sorry, I'm done there. All right. Now, lastly, on genetics is this question of the dihybrid cross. Identify what is Z. You just have to know. Cross this, and then you write what is Z. What is the genotype of the parents? Now, how do I identify the genotypes of the parent? Genotype of the parent. Look at the gametes. You'll find the genotypes from there. Start with F. For example, you write the F. If it is repeated, leave it. Is there any other small f? Yes, the small f is there. Then go to capital H. Yes. Is there any other small h? Yes, is there. Done. Look at them. If there was no, if all of them were capital H, ne? All of them were capital H, ne? For example, capital H, even here capital H. I would have said, do I have F? Yes. Do I have small f? Yes. Do I have capital H? Yes. Do I have small h? No. They are all capital H. Done. That's how you can identify. Simple, no stress. They're saying that number of the genotype that could result in offspring that is short finger, and short is dominant, ne? And then this one is recessive. Because it's recessive, I need to have with the small h. As long as I have capital H, is 1, is 2, is 1, is 2, yours, and then yeah, is 3. So there are 3. And then allele that for continuous hairline, allele, not genotype. So it's gonna be small h. The genotype of the child that is heterozygous recessive for both alleles. So is this one, which is small f. Yes. So this dihybrid cross, simple. You should not go to the exam when you don't know anything. Welcome to evolution. Now, evolution. Okay, evolution. Let's look at evolution by natural selection. This one does not have too many questions, which I'm gonna speak about, but it has many questions in human evolution. However, these days we are setting evolution by natural selection than human evolution. I think Mutasi, now we are on you now. Ne? Share the video so that now people can join for evolution. Ne? Because they find a lot of challenges with the evolution. Evolution, what is a hypothesis? What is a theory? You have to go when you know the difference. Ne? Yes. Hypothesis basically is a tentative statement which is subjected to be tested. It can be proven correct or wrong. And then a theory, basically what you need to look at is a statement which has been proven by facts. You understand? Situation which is being proven by facts. So it means that it must have evidences to support it. Then it becomes a what? It becomes a theory. Now, before I go there, I think this one is a little bit clear. The 6-mark questions or 7-mark questions, they are going to come from here. Number 1, theories of evolution. Number 2, that is Lamarckism or Darwinism. Number 2, speciation. Number 3, present day evolution. Now this difference between Lamarck and Darwin, yes, but present day evolution. You can't go to the paper without, because these are six marks. 6 marks each. Yeah, I told you, you go back, you come back and then you tell me what you found there. Ne? Yeah. All right, let's continue. Theories of evolution. Lamarck's theory, Darwin's theory. Lamarck, what you need to know is how to apply it, but I'm just going to talk about it. Ne? I'm not going to take too much time on it. I will spend the last few minutes on evolution, human evolution. But I'm going to explain everything so that you don't lose anything. Lamarck's theory. State 2 laws of Lamarck's theory. It's law of use and disuse. That's number 1. Number 2 is law of inheritance of acquired characteristics. So it means that Lamarck has 2 laws. Number 1, law of use and disuse of a structure. Number 2, law of inheritance of acquired characteristics. Why is Lamarck's theory not accepted scientifically or by scientists in the world? Number 1, there is not enough evidence to support his theory. Number 2, organisms don't change because they want to change. Organisms change not because they want to change. You understand? Yes. For him, he says that if you want to be tall, you just become tall. If you want to be short, you just become short. No, organisms don't change because they want to change. Number 3, what is the difference? You have to know how to use how how Lamarck's theory is applied. For example, how did the big elephants with the big trunks came up with the big trunks using Lamarck's theory? So meaning that the more you use the structure, the more. At the beginning, all elephants had short trunks. Then you tell us that these elephants, they use their trunks frequently, maybe to defend themselves or to reach the trees, and then their trunks became bigger and longer. They passed their genes. Yes, they produced babies with big, long trunks. But if you are using natural selection, let me just use this as for natural selection. You have to start with, there is variation among the elephants. Those with long trunks and those with short trunks. Those with long trunks, they could survive. Yes? Why? Because they are able to defend themselves. They could reach the tall tree plants. While those with short trunks, they could not survive. They died in large numbers. You understand? This is called natural selection. This is called natural selection. I think. Now, those with big trunks, they pass their genes to the next generation. After many generations, all elephants had big trunks. Let me show you. Let me see if we can get any question here. But you can't go to the exam without knowing that. Using sex, show gender in human, how it is. I see. I explained this. I explained this. You see, 6 marks. 6 marks. Okay, when I find it, I'll try to show you that example. Speciation. Now, students confuse speciation with natural selection. What is speciation? Speciation is the formation of new species. Please don't confuse those two. Look, speciation is the formation of new species. What's a species? It's a group of organisms that can interbreed, meaning that they have similar characteristics. Group of organisms with same characteristics that can interbreed to produce a fertile offspring. What is the population? The population is the group of organisms of the same species that can interbreed to produce a fertile offspring. That's it. Now, speciation is the formation of new species. When they talk about speciation, they will bring about, for example, one area, and then this area is divided into 2. You understand? And then after that, if you try to mix them, they will form different organisms. They will form different organisms. They will form different organisms. So now, let me explain to you. You have a population of A. So you have to tell us that there was one there was one population. Yes. Now the population is divided into 2 by a geographical barrier. You have to tell us the geographical barrier. Let me show you, because I saw a question here. Yup. So what is the geographical barrier here? The geographical barrier describes speciation of poto and lemurs. How did they become different species? There was one population of these organisms. There was one population of these animals. Did they talk about the ancestor? If they talked about the ancestor, then you have to speak about it. Scientists believe that poto and lemur share a common ancestor. They didn't mention the ancestor. So you have to tell us that. Don't say that there was one population of poto and lemurs. They have not yet become poto and lemurs. No. So they have not yet become those new species. You have to start with what they started with. There was one population of these animals which shared a common ancestor. Yes. And then what happened? The population was divided into 2 by 2 subpopulation by a geographical barrier. What is the geographical barrier here? Ocean. So you don't need to tell us by geographical barrier. Tell us by the ocean. If it is a river, tell us by the river. Unless the question is just open. And then now, you formed 2 subpopulations. That is east coast of Africa and Madagascar. And then what happens? There is no gene flow. There is no. This one can't come this side. Gene flow means that there is no sex between them. There is no gene flow between these populations. Whereby these 2, they experience different environmental conditions. Are we together? So because they experience different environmental conditions, natural selection occurred differently. Oh, someone is asking me about punctuated equilibrium. I will come there. They formed, I'm going to explain it. Different environmental conditions, therefore natural selection occurred differently. Whereby they became genotypically and phenotypically different. And if you remove the river, if you remove the ocean, they cannot mix to interbreed. Therefore, they formed 2 new species of poto and lemurs. They formed 2 new species of poto and lemurs. You can only talk about these 2 new species. Don't talk about 2 species before you talk about genotypically and phenotypically different. If you talk about it, you're gonna lose marks. you're gonna lose marks. Even if someone is marking, it will pain, but nothing we can do. You will lose the marks. So what do? Is you only talk about different species after talking about genotypically and phenotypically different. If you don't do that, and you talk about you form two different species, that the population was divided into two different species by the ocean, you are gone. You have lost the marks. The paper has killed you. You didn't kill it. I think you understand what I'm saying. Yes. So now, I repeat. Let me repeat it for you. There was one population of a common ancestor. One population of common ancestor. The population was divided into two subpopulations of Eastern Coast Africa and Madagascar by what? By the ocean. There is no gene flow between the two subpopulations. You see, they are still populations, not yet new species. Remember, a population is a group of organisms of the same species. And then now, they experience different environmental conditions. Natural selection occurred independently or differently. And then now, they became genotypically and phenotypically different. If you remove the ocean and you let them mix, they cannot interbreed to produce a fertile offspring. Therefore, two new species of Poto and Lema have formed. They can bring when they are more than two or more than three. So in this case, you tell us three new species have formed. So don't stack on two new species. Don't stack on one new species. So two new species. So I'm coming for punctuated equilibrium. Charlotte, yes, you are saying hi. Are you still here? All right. Now, let's go to punctuated equilibrium before I go to present day evolution. Oh, let me just finish the present day evolution and then I'll talk about the punctuated equilibrium. If I forget, you remind me. Present day evolution, it deals with the bacteria, virus, black peppery moth. We are talking about present day evolution. Evolution which is taking place, which we see. You understand? DDT on mosquitoes. Yes. So, you know, if they are bacteria, it means that you're going to use antibiotics. Use the same principle of natural selection. Please don't confuse natural selection with the speciation. So in this case, you will say that there was one population of bacteria. These bacteria are separated by the variation which is there. So now when you're explaining, you say that there was one population. Sorry. You say that there was variation among these bacteria. Those which are resistant to antibiotics and those which are not resistant to antibiotics. Then now tell us that the bacteria which are resistant to antibiotics, they survive on the application of the antibiotics. While those which are not resistant to antibiotics, they die. Those which are this is called natural selection. Those which are resistant, they pass their genes to the next generation. So the next generation will have more bacteria which are resistant. And after many generations, the antibiotics will become ineffective to bacteria. Let me repeat. There is variation among the bacteria. Those you can use for mosquitoes about DDT. You can use for virus about the antiviral or ARVs. There was one. There is variation among the bacteria. Those which are resistant, you have to tell us the variation. If you don't tell us the variation, you lose the mark. There was variation among the bacteria. Those which are resistant and those which are not resistant. Those which are resistant and those which are not resistant to antibiotics. Those which are resistant to antibiotics, they survive on the application of the antibiotics. While those which are not resistant, they die in large number on application of antibiotics. antibiotics. Those which are resistant, they pass their genes to the next generation. After many generations, the bacteria are going to become ineffective to antibiotics. Simple. There is no way how you're gonna skip. Oh, they're gonna bring a lot of things which you don't know here. Never. What is the difference between artificial selection and natural selection? And what is the difference between Darwinism and the Lamarckism? Difference between artificial selection and natural selection. You know that artificial selection, what is basically on artificial selection is what is the selective force? First ask yourself, what is the selective force? Selective force in artificial selection is human, while in natural selection, the selective force is nature, is the selective force. You understand? Yes. And then Lamarckism and Darwinism, the major difference is Lamarckism, organisms change because they want to change. But in Darwinism, organisms change due to the environment. Yes. Then number two, in Lamarckism, individual in the population change. Like if me, I want to be big, I become big. If I want to be small, I become small. Not the whole population. But in Darwinism, only the whole population change. Remember, when Corona came, if you want or not, they say that you have to put on a mask. Natural selection. You don't put on a mask, you die. Whether you want or not, you have to do so. You understand? Therefore, that is natural selection. Is it clear? So, in Lamarckism, individual in a population change. While the Darwinism, only the whole population will change. So you can also talk about other differences between the two. Let me talk about punctuated equilibrium. And then after that, I go to human evolution. Someone is asking me, how do I draw a pie chart? How do I draw a pie chart? I'll go to the last question. That is the last question, question 3. The pie chart, how do you draw this at the end of human evolution? So that just I show you how do I identify independent variable, dependent variable. How do I get it from the question? How do I know the control variable? How do I know the conditions which are supposed to be put in place before you start? All right, punctuated equilibrium. It is very simple. Gradualism versus punctuated equilibrium is gradualism versus punctuated equilibrium is saying like this. You have two scenarios. Punctuate gradual. When you say gradual, it means that it's slow. The change is slow. While punctuated equilibrium, the change is fast. You have periods of no change and periods of change. The change is rapid. Periods of no change, periods of change, periods of no change, periods of change. This is punctuated. Punctuated. What this is gradualism. Here you require a lot of time. A lot of time. Here it occurs in short periods. So punctuated equilibrium, you have periods of no change, periods of change, period of change, period of change. And in most cases, the punctuated equilibrium is caused as a result of mutation. While this one is changed, is caused as a result of natural selection. Yes. Natural selection here, yes. While here, mutation. Why? Because you just wake up in the morning and then you have something different which you didn't have before. Mutation maybe occurred. So that is punctuated equilibrium, or the other side is gradualism. This marks the beginning of the end of evolution by natural selection. Let's kill the human evolution now. Human evolution. Human evolution, guys, is The most important part where students sometimes lose marks is a phylogenetic tree. I don't know why, but we'll talk about it. Someone is asking, do you do calculation for pie chart? Yes, you do the calculation for pie chart before anything else, because you can't do percentage in a pie chart because the circle is not in percentage. All right, let me just see if I can find here a question. The first question in human evolution can be, to which family do humans belong? To which family do humans belong? They belong to family Hominidae. 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 Differences between, let me tell you what are the short questions. Short questions, guys, in human evolution. The short questions which you're gonna find, and then you have to give the answers. 6 marks. Number 1, it is out of Africa. Hypothesis. That's number 1. Number 2 is, so here you have to tell us the evidences. Evidences of that is fossil. You have to tell us the mitochondrion, DNA. Sometimes you call it genetics. Then you have to tell us cultural evidence. And then they can also talk about the differences between humans and apes. Yes. And then also they can talk about similarities between apes and humans. There is no 6 question mark they can bring here. Nothing. I repeat, nothing. Unless it's gonna come from heaven. Okay, now let me explain. There are some things which we changed a bit, and you have to know. That's why if you check my book of distinction material, you'll find out that it's always updated each and every year. It's not static. It's always a version for that year. There will be a version for 2023. All right. Now, let's continue. They're saying that, now look at this. Position of foramen magnum. They're saying, which diagram represents the skull? There is a difference between a skull and a cranium. A skull is everything which is encloses the head. That's the skull. But a cranium is only the part which encloses the brain. Only the part which encloses the brain is what you call cranium. But a skull is everything which encloses the what? The brain. I think I'm clear there. Number 2. Number 2, they're saying that which diagram represents the skull of a bipedal? Now we have 3 factors which 3 factors which affect or which do the bipedalism, of which they can ask you the advantages of bipedalism. You can see far, you can hold the organisms, you can hold the objects. Sexual dimorphism is exposed. You can see the sex organs. They can be shown because you are walking, yes. And then the body is lifted. You lift the body out of the ground, yes. So it means that less heat is gonna be reflected to your body. There is small surface area for heating of your body from the upper side. So those are some of the advantages of bipedalism. But now, what are the factors which contribute to bipedalism? There are 3. The first one. Why is this thing doesn't want me to write? I will write even if doesn't want me to write. Okay. Number 1 is shape. You look at the shape of the spine. Shape of the spine is what? Is shape. And then you look at the pelvis. Pelvis is short and wide. And then number 3, these are factors which affect bipedalism. Short and wide. Is wide, ne? And short. So if you look at it, is wide and short. And then the third one is the third one is the position of foramen magnum. It is in forward position. Ne? Yes. Now, before I go to explain this, what is bipedalism? We changed this. You don't just say that bipedalism is walking on 2 limbs. Which limbs? Hind limb or forward limbs? You have to tell us walking on 2 hind limbs, or walking on 2 lower limbs, or walking on 2 feet. But we prefer walking on 2 hind limbs. If you're using limbs, it must have the word hind limbs. So, how is the shape of the spine able to for bipedalism? Shape here is S shape to allow flexibility. That's number 1. And then pelvic girdle is short and wider to support the upper weight. Position of foramen magnum is in forward position to allow the spine connect vertically. So basically, that's what you are supposed to tell us. Then they are saying that the next question is tabulate visible. They are saying visible. Don't bring about anything else. Visible difference between the upper jaw and the lower jaw of A and B. Check upper jaw, lower jaw. They're only looking for this. You see. In most cases now, when we bring these, you have to know the differences. So in this case, you talk about the canines here are well developed. They are not well developed. There is one student who is saying that this one has canines, this one has no canines. No, the canines are there, but these are well developed. Then this one are less developed. This one is rectangular shape. The jaw is rectangular shape. While this one, the jaw is what? Is C shape or U shape. C shape. Then if you look at here, you have spaces between the teeth. There are spaces between the teeth, large spaces between the teeth. Here, no space between the teeth, or small space between the what? Between the teeth. Now, they can ask you, now I'm going to start firing you questions. How is the brain size related to intelligence? How is the brain size related to intelligence? The brain size related to intelligence, or how is the skull related to intelligence? How is the brain size related to intelligence? If the skull is big, it is going to accommodate more brain cells. The higher the brain cells, the higher the intelligence. If the brain size is big, you have more brain cells, and then this is linked to intelligence. That's why the previous organisms, for example, Ardipithecus, they had a small skull, therefore they had a small brain capacity, therefore the intelligence was also small compared to us. And when you are explaining this, when you're explaining the brain, there is something which you are looking for. If you don't mention it, you don't get the mark. The brain of human is not bigger than the brain of the elephant. It's only bigger than the brain of the elephant if you are comparing it with the body size. If we have a big brain compared to the body size, compared to the elephant. So when you are comparing, when you're talking about the brain or the cranium, you have to talk about compared to the body size. Please, when you talk about cranium, comparing them, then you compare it with the body size. I think I'm clear there. Who discovered Homo sapiens? You have to know who discovered it. I think I should drop. We have a short video which is talking about it. Who discovered the Homo sapiens? Tim White. Oh, who discovered Ardipithecus? Tim White. Tim White is Australopithecus. Sorry, is Homo sapiens. Now, who discovered humans? Is Tim White, yet is also Homo sapiens. He discovered himself. No, he discovered the oldest skull of Homo sapiens. Is it clear? Yes. So, you need to know, don't go to the paper without knowing 3 species discovered in South Africa. South Africa, at least 3. Don't go without knowing the 3 species which belong to Australopithecus africanus. Please don't. Which is Mrs. Place, Town, Child, and then Little Foot. These are the 3 Australopithecus africanus discovered in Africa. What is the transitional fossil? A transitional fossil is that fossil which has the characteristics of the ancestor and the next generation, or the children. It means that it has not got extinct. It's still in transition. You understand? Which one of the following is a transitional species? In most cases, we look at Karabo. Australopithecus sediba is the transitional species between Australopithecus africanus and Homo habilis. So that is the only transitional species or fossil we talk about. However, even Homo naledi is also a transitional what? A transitional fossil or species. Now, question. Give the difference between the skull of humans and other primates. There is something which I want to talk about prognanthus, because when you are marking that one, humans, prognanthus means protruding jaws. So the other primates, they are prognanthus. Humans are not prognanthus. Don't say they are less prognanthus. No, if you have it in your book, yes, I know it's there, but we will not give you a tick. Humans are not prognanthus. Protruding, ne? Yes. While the other ones are prognanthus. Flat face, sloping face. Those ones are fine. Foramen magnum in forward position. We don't want anything else. Forward position. Foramen magnum in backward position. Don't tell us that foramen magnum is in the center. Center where? Foramen magnum is in forward position. Foramen magnum is in a backward position. Those are the things which changed, and then you needed to know this before you go for your paper. Because if you make a mistake and you mess them up, you're gonna lose a mark. Yet you knew the answer. It's just to rectify those few small mistakes. Lastly, describe, state the out of Africa hypothesis. State the out of Africa hypothesis. We say that modern humans, the word modern must be there. Modern humans originated from Africa. Modern humans originated from Africa and migrated the rest of the world. Originated from Africa and migrated. If you don't write the word modern, it's gone. The mark is gone. Give 3 evidences to support the out of Africa hypothesis. That is fossil evidence, mitochondrial DNA, and cultural evidence. Now, remember I said that this can come in this 6 mark. Yes? So how do I use cultural evidence? How do I use the mitochondrial DNA? How do I use fossil evidence to do this? Now, fossil evidence, you need to talk about few aspects. You have to talk about Adipithecus. You have to talk about Australopithecus. You have to talk about Homo habilis. You have to talk about Homo erectus. You have to talk about Homo sapiens. This is what you're supposed to talk about. Tell us that Adipithecus, and then Australopithecus and Homo habilis only found in Africa. I think. Then when you reach here, you say that oldest, and also when you reach here, you say oldest fossil of Homo erectus and Homo sapiens were found in or discovered in Africa. I repeat. The fossil of Adipithecus, Australopithecus, and Homo habilis were only discovered in Africa. Only in Africa. Fossil of Homo erectus and Homo sapiens, the oldest fossils were discovered in Africa. That's how you're supposed to write the answer. Now, what about mitochondrial DNA? Here in mitochondrial DNA, we use the mutations which occurs in a mitochondria. Why do we use mitochondrial DNA? Because it has a small DNA, so it is easy to focus on it. But it's rare to find mutations there. So if the mutation occurs, we can use it to trace the lines of what? Descent. And if we trace them, it brings back to Africa. That's why it's called the mitochondrial Eve. And then cultural evidence, you talk about the oldest tools were discovered in Africa. And those are called the Odowan tools. Odowan tools. Yes. As I'm winding up with the today's class, so that you go and get your distinction, please, if it requires you to repeat this, to rewatch it, I'm telling you, I've tried to discuss each and everything which you're gonna find in the paper. You understand? If I've forgotten something, it might be only few, but you have 90%. I'm saying 90%, meaning that, what does it mean? It means that over 130 marks, you have the marks. All right. Let me conclude with the pedigree. Then I'll just talk about few of the experimental question, and then lastly I'll go to the terminologies. Just only talk about the terminologies, and then we conclude for this class. I thank everybody who is attending, who is in my class, and I wish you the best. You must get a distinction. Let's try to finish up. Now, pedigree. Not pedigree. Phylogenetic tree. Before I go to phylogenetic tree, how do you write the scientific name? I think phylogenetic tree I need to use a diagram here. Let me just put out the diagram here, which I can use. Okay. All right. Let me use this. It's fine. Let me check if I have any phylogenetic tree here. This is the thing I was trying to explain before. Describe one other structure difference in bipedal and quadrupedal. You can talk about. Oh, okay. I gave the difference, ne? Short and wide, the factors which affect that. Okay. Pelvic girdle. You have this now because I tried to explain it. Okay, let me use this. I'm gonna tell you all the questions which can come here. How many genera are indicated here? How many genera? Genera comes from the word genus. How many genera? Here is it's getting to 6 o'clock. To 6 o'clock. I don't know whether the electricity is going this side. Ne? Just in case that it has gone, at least you have everything. Ne? Yes. When you see me offline without saying bye bye, it means that it has gone because I didn't check anything. Describe the process of natural selection. Now you see this one. You just explain. You just explain. You just explain. Now I explained it. You don't use any example. Ne? Yes. Okay. Now name. How many genera are indicated here? Genera is genus if it's one. Genera when there are many. You count the first name, but you don't repeat them. For example, 1 Australopithecus, Homo 2. Do I have any other? Nothing. So it means that I have only 2 genera there. To which family do humans belong? To which family do all to name the family to which all the represented organisms belong. To which family do we belong? We belong to Hominidae. Yes. Then they are saying describe the cultural evidence used as a tool. You see, I explained it. Yeah, oldest tools are other ones. Ne? So it means that how does it support the out of Africa hypothesis? It supports the theory of human evolution. Simple tools were discovered with the simple skulls, and then which means more advanced tools with the more or larger skulls, which means that they had the ability to modify these tools and were found in the upper layers. How long did okay, now let me start. How long ago did most recent common ancestor of this and this existed on Earth? Now, when they say how long ago, for example, did the Homo ergaster existed, you come here. For example, if this is Homo ergaster, I come and draw where it appeared and where it got extinct. So it means that I will say 2 minus 1, which means how long ago? No. How long ago did they appear? This is how they appeared. It means that it's going to be 2 million years. How long ago did they extinct? It's going to be here. So it's going to be 1 million years. How long did they exist? How long did they exist? That's a question. It means that I'm going to say where they appeared and where they got extinct, and then I said 2 minus 1, I get 1. But here is going to be million years. It's not going to be million years ago. It's going to be million years. I think I'm clear there. Here you say million years ago. Here you say million years. Sometimes we always mark this unit. Then now they are saying which species first used the tools. When they ask you which species, if it is not indicated here, it is Homo habilis. That's why it's called handyman. Which species first walked on 2 limbs? If they ask you species which first walked on 2 limbs, if it's not indicated here, it's gonna be Homo erectus. I repeat, Homo erectus. Then, which species are competed for resources? In this case, you'll find out that if this species and this species, they exist at the same time. If you draw a line like this, you see that this one and this one, they touch each other. It means that they exist at the same time. It means that they competed for resources. So maybe you'll say that Homo ergaster and Homo habilis. So it depends on the species which existed at the same time. But it must be having a line where they touch each other. Not exactly, but they must have a line where they touch each other. Then now, they are saying, what are they saying? They are saying, explain the possible reason why Homo ergaster was placed between Australopithecus afarensis and the Homo. It means that this is a transitional fossil. Yeah, it's a transitional. It's a transitional fossil, meaning that it has the ability, has the characteristics of this organism, and then it has also characteristics for this. All right, explain the possible reason why Homo ergaster. Where is Homo ergaster? Homo ergaster is here, and then Australopithecus afarensis is here, and then Neander is here. So it means that this is a transitional fossil. Yes, it has this one. Homo ergaster has some characteristics of this and also some characteristics of that. Because it didn't get extinct, therefore it's still going. Therefore it is a transitional fossil. How is the brain size compared to intelligence? I explained that. There's no questions they're gonna bring about this and then you can't answer. Yes. Lastly, how do I identify independent variable and dependent variable? Those who attended my paper 1 class, I think you know exactly. Just write ID. ID. And then after that, X, Y. I think. After that, you tell us what is ID. Independent variable, not identity card. Independent variable, independent variable, independent variable on x-axis. So when you're drawing, independent is x-axis is here. So independent variable must be here. It must have units, please. Then this one, dependent variable, is this y-axis. So dependent variable is here. And then now, please put the title. It must have both the independent and the dependent. Put units here, please. Don't forget about that. And then now, because if you forget, you lose the marks. Scale, please. Don't say 2, 4, 7, 8, 10. No, the range is on the same. Here is 2, here is 3, here. You have to use the same scale. 2, 4, 6, 8, 10. You understand? If you miss them, you lose this and this. And then now the problem, now you have got to know that independent is x, dependent on y. But how do I identify them from the table? In most cases, the first column, however much, how many columns you have, is ID still. The first column in most cases is independent, and the second column is dependent. Please don't miss up. Yes, the second and the third is always dependent. So now, how do I plot? How do I plot, and how do I draw a pie chart here? If you are drawing a pie chart, please convert this into degrees. For example, if I have maybe 10, 20, 20-ish. No technology. 20, 30. For example, this is Africa, Asia, maybe Europe. Then they say, so to put this on a pie chart, you have to say 10 plus 20 plus 30. So it gives me, this is 50, 60, 60. That's the total. So to do this, I'll have to say 10 out of 60 times 360, because this is the pie chart, pie chart, which is 360. So it give what you get here. You do that for 20, you do that for 30, then you plot. Protractor measure for this, then you change the protractor, start from here now, measure, then the remaining one will be for that. Then you put the title, then you shade whatever you shade, you put a key here, you get the answer, get the marks. So please don't mess it up. And then now, that is a pie chart. But now there is one for line graph. If it is on the same axis, and then the bar graph, it is if it is a bar graph, yes. For example, you have 20, 30, 40. Ne? Bar graph, you plot this and this, you get that and that. Then plot this and this. Then after that, give a space. Ne? Plot this and this, maybe 20. And then plot this and this, maybe 50. Then a space. Now, these bars must be the same. If this bar is not the same as this, you lose a mark. If this bar is not the size must be the same. These spaces must be the same. Ne? If those spaces are not the same, you lose a mark. That's how kids lose mark with these graphs. Then, if it is a line graph, it is a little bit different. Because a line graph, you have to first plot one by one. Line graph, look. Line graph, this and this. But in most cases, to get a line graph, you have to be with the figures, maybe 10, 30, 40. So that this and this, you plot, you get a tick there. This and this, you plot, you get a tick or a spot there. This and this, you plot, you join. Then if it is two graphs at the same time, now this and this now. So you first finish one column and another column, but independent must be kept. Now this and this, then you plot. This and this, then you plot. This and this, then you plot. Yes. Now, how do I know that my graph is wrong? We don't have a graph which runs like that. Some people have, it means that you swapped the axis. If you still have time, redraw it. We don't have a graph which runs like that. No, this is not met. Yes. So the graph must either be straight, going If it's going up, it must form a wave, or it's going down, but this so-called parabola, what? No. We don't have them. Once you see that, you just know that you have swapped the axis. It means that what was X must be Y, what was Y must be what? Must be X. Please take note of that. Last